So in this recording, we want to address what's called as or what's known as the dogma of molecular biology. And dogma is anything that is unquestionable. Oftentimes you'll hear it in reference to church doctrine. This is something that you cannot question. It is always true. And we know, of course, that that's absolutely absurd. There's nothing in science that we can't learn more about and that can't change. So dogma is not a true term for molecular biology, but we're going to use that because this idea of dogma, the idea that information in DNA is transcribed to RNA and then translated into proteins, and proteins do things, is so central to how all of biology works that we call it dogma. And so this flow of genetic information is what we refer to. Now, what this is going to also do is link two important terms that we need to understand, and that is genotype and phenotype. And so genotype is the information in the DNA, and phenotype is the physical manifestation of that information. So whereas you have genes that encode for proteins that make your eyes brown, you would say the gene that you have is your genotype for eye color. And then the fact that I can look at you and see that you have brown eyes, the fact that you physically have brown eyes, is your phenotype. And so those genes are expressed, we talk about gene expression, to achieve a phenotype. So we talk about genes being turned on or turned off, and this is gene expression. We'll go over this more in great detail as we move forward. So as we begin to study this, and we'll look at some of those early experiments next, we realized that DNA did encode for proteins, and it was thought that there was a very linear kind of relationship. There's a gene equal to a protein equal to a trait, and we find that it's much more complicated than that. So we're going to see things and study stuff called polygenic inheritance, pleiotropy. They're going to argue against this very simple model, but the model is so fundamental, it's a great place to start to truly understand how DNA works. And to do that, let's do a little bit of history, and we'll take a look at some kind of a classic geneticist approach to understanding DNA. And for a geneticist, it's all about finding mutants. Something that was working and now is broken, or is different. And then if you can look at what's broken and look at the results, you may understand a little bit better how something works. So if we look at the classic example of Beadle and Tatum's neurospora work from the 1920s, we can talk about how they started to build this kind of one gene, one protein hypothesis. So what they did was they took a fungus called neurospora, and he grew it on what's called minimal media. And so this media is like jello, and you see it here in a test tube, and that brown line on the top of that jello is the growth of the fungus. Now, if you irradiate or mutate the fungus, you find many strains of it then that don't grow on minimal media. They can no longer make some of their essential nutrients from the minimal media. Something is broken. And so what you can see is that you can start to classify the ones that can't grow into different groups. And this is what's called mutant rescue. So by adding back certain components into the media that weren't there originally, that normally the wild type or the most common form of the fungus can synthesize on its own, you can rescue the mutant. It can grow again. So for example, the class 1 mutants, if you add ornithine back, you get growth. Class 2, you add citrulline back, you get growth. Class 3 mutants, you have to add the amino acid arginine, and you get growth again. So these are all steps along the biosynthetic pathway to make the amino acid arginine, which is an essential amino acid. You need it for your proteins, and we all know that proteins do stuff. So if you don't have these proteins, it doesn't work. So what they then proposed that was what was broken in their different classes of mutants were genes along that biosynthetic pathway. And so by adding back the product of that gene activity, so an enzyme is going to catalyze a chemical reaction. In this case, take the precursor to ornithine in class 1. In class 2, take ornithine to citrulline. In class 3, take citrulline to arginine. And so you have to always add back what the mutant can't make to save it. All right. So you're going to want to have to think about this example a little bit to kind of understand how that works, and we're going to come back to this idea again and again, so I want to introduce it now. But these experiments really established a pretty strong paradigm in the field, this idea that you had one gene, one protein. Right? But then we discovered much later, many years later, that in fact we're going to have one gene produce many different proteins. Okay, and this is going to be something that's very specific to eukaryotes. As complex organisms, we're going to see that our genetic information is much, much richer, basically, than prokaryotes because we can make many proteins from one gene. Okay. So this idea of dogma, what we want to talk about are the basic principles and outline this in a general sense, and then we'll go back in future recordings and discuss it in greater detail. So basically, we have RNA as an intermediate between DNA and proteins. The process of making RNA from DNA is called transcription. The result of transcription then is this messenger RNA, and then that messenger RNA can be used in a process called translation, which is going to enable us to synthesize the polypeptide from the information in the mRNA. So ribosomes are going to be the point of this protein synthesis. So we can see this in a couple of cartoons. First, in a prokaryotic cell. So we can see that the DNA is out there, free in the cytoplasm. We're going to get a transcription event and get an mRNA. That mRNA then can be bound to a ribosome and read, quote unquote, to produce a polypeptide. Now in a eukaryotic cell, we talked about eukaryotic cells being our mansions of cells. They have many, many rooms. So we have different rooms to sequester different parts of this process of the dogma in the eukaryotic cell. And so in the nucleus, we're going to have transcription occur, and we're going to get an exact copy of the DNA in what's called a pre-mRNA, or a primary transcript. That pre-mRNA has to be processed. Again, what does that well? The nucleus does it well. That room is made to process the primary transcript to the mRNA, the mature mRNA, and that mRNA then is going to exit the nucleus and engage ribosomes, which then make the proteins that do stuff. So again, the dogma of molecular biology, the foundation of how DNA works, is that the information in DNA is read, is copied into an mRNA molecule, and then the mRNA molecule, the information from that, is read and used to make a polypeptide. And proteins, of course, do stuff.